All right. Okay, it's a great pleasure to have Jure visiting us from Famous Academy of Sciences in Beijing. Jure has spent several years working on many soft matter problems, including self-assembly. And today he's going to talk about his title of the topic is one, but multivalent binding in molecular targeting, recognition, and activation. So he is here till today or tomorrow.
Tomorrow mornings.
Tomorrow morning, but we wanted to talk about the over to
Thank you very much. Since I talked about our work on virus capsid assembly and genome packing in Edinburgh, I decided I would present our work in the recent years on multivalent binding here. So basically what I'm going to talk about is this kind of systems. Let's say you have a surface or typically cell membrane which is decorated with different kinds of receptors, proteins on its surface, and you want to target this, meaning you bring some particle or some other cell that binds to the surface. For many reasons, many obvious reasons, this is very desirable to be able to target specific cells. What we wanted to explore initially, all the time that we are working on this project, is how to target selectively or super selectively. So meaning that you want to target cells which are similar but slightly different, and you want to only target one type but not the other. That is the key problem that we worked on. And then also as a next step, maybe also think about not only binding to the cell membrane, but also if these particles then can be endocytosed or go into the cell. But mostly we are concerned with the first step of targeting, binding to the surface. This kind of problem has been already quite well resolved in case of targeting bacterial cells. Namely, antibiotics do this perfectly well. So if you have on one hand bacterial cells that invade your body, and all the other cells are your own human cells, this task is in principle much easier than what I described before, because it turns out that bacterial cells express some receptors that are not present in human cells, or almost not present in human cells, and then in a very simple explanation is that antibiotics would target those receptors, and they can target them with specific antibodies, specific molecules that only bind to that receptor, and that works well. And this is a successful selective targeting. But what we want to explore now is a different problem, and namely, if in our body we have cells that have a certain receptor expressed in some to some degree, and other cells, like for instance cancer cells, overexpress this. So we have cells that express the same receptor but in different quantities or different densities, and we want to target like here. So we want to target these cells which overexpress a certain receptor and not these cells. Now, if we have the same approach as the antibiotics, traditional pharmaceutical industry keeps doing, that we search for specific antibody that strongly binds to this kind of receptor, yes, that we want to target, then you see that if this receptor has a strong affinity to, if this ligand here has a strong affinity to these receptors, it will bind to the cell no matter if it's like a sparsely populated or densely populated with this receptor. And this is the problem with traditional approaches like chemotherapy. This is a scan of the body of a patient, and the tumor is located here. And then this is the scan of where the drug ended up after the chemotherapy. And you see that it is successful. It goes to the tumor, and it presumably then is able to kill the tumor. But you see, it goes to many other places in the body and even to larger extent. So the problem with this is that the chemotherapy kills the tumor, but also kills the patient. So you have to be very careful in dosage. So you have to use a very small dosage, and then this is not so efficient. So the dream scenario would be to have the drug that only binds to this receptor, to these cells, and does not bind to the cells that have a different composition. And then you could use stronger dosage. It would be targeted and much more efficient. And that is the harder problem than targeting different receptors now, like antibiotics. Now, if I say super selective, what do I mean? Typically, our system that we studied is like a particle that has several ligands on its surface, and then there's a surface with several receptors, and the ligands can bind to the receptors. Now, if we have several particles on a large surface, we can calculate the coverage, surface coverage of the surface, and plot the surface coverage, let's say, as a function of the density of the receptors. We studied different surfaces with different density, and now we can have different scenarios. Let's say if we have the dependence which looks like this blue curve, this is not selective. We increase the density by several orders of magnitude, and the surface coverage only goes up by a small factor. That we call non-selective, and what we call selective is this kind of on-off response. Yes, there is practically no binding until a certain threshold, and then it jumps to complete coverage. So this is something we look for, and if we plot this sensitivity, selectivity parameter, is just the slope of the logarithm of the coverage over logarithm of concentration. If the coverage scales like power law, then this selectivity parameter is 1 for linear dependence, and that's not selective, and it's much larger than 1 in the selective case. So a super selective case would be this when the selectivity parameter suddenly becomes much larger than 1, and that's the system we want to design. So just one comment: if we have this kind of system, this coverage, this looks like very much like Langmuir adsorption. The only difference, an important difference, is that in this is activity. This is a partition function of a single ligand binding to receptors, but there is this dependence on the density of receptors, which is not there in the simple Langmuir adsorption model. So, and this makes a difference. So this is what makes it a bit more interesting. Okay, so now the first work on this was done by Francisco Martinez Veracoechea, who was a postdoc of Don Frankel, and he explored this, compared, like, now how could you achieve such a super selective response. And first, if you take monovalent binding, so that each particle can form a single bond to the receptor, and its bond is either on or off. Then you would assume that, okay, you can change now only the free energy of the bond, yes. And if you go to large, stronger bonds, you cannot increase the selectivity. You increase the binding strength and you increase the coverage of the surface, but you don't increase selectivity of this coverage to the density of the receptors. So in fact, if you go to stronger bonds, you actually decrease the selectivity. Yes, you become less selective. You bind more, but you bind more to everywhere. So the conclusion is that you cannot achieve a super selective response with monovalent bonds. No, that's impossible. If you want more responsive system, you have to go to multivalent. So each particle has to make more, have to be able to make more bonds with the surface. And so here is now a model with 10. Yes, K is the number of ligands on each particle. And you see in case of 10 now, if you increase the, if you decrease, this is the very strong binding case, the selectivity is still not great, but if you then go to weaker and weaker binding, you get more and more selective response.
Sorry, some simulation or is this sort of the analytic model?
This is the simulation, but it is very much, you can get this behavior with the analytic model that I showed you. This is the partition function, and you have a model for this partition function. If you assume independent binding, I mean, you have to make some assumption, and you can do an analytic model which very much agrees with this. Sure. Yeah, super selective. I mean, it's very selective. Like it's just a term that Francisco and Dan came up with in that paper. It's like it's like the perfect super selective case would be on off, like 0 and 1 as the step function. And if you're very close to that, you call it super selective. Yeah.
The more negative it is, the more binding it is.
Yeah. 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 Well, of course, if it's positive, then you don't have binding. So we only study negative here. Huh? Yeah. 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 So you get this. Let's say. I would call in this, like, it's not a very clear, very rigorous definition, but I would call this super selective, and the alpha goes like to, which becomes much larger than 1.
But you're at beta, it's changing sign. So
where?
So when the next line, take that next time here, go from -1 to 7. That was my question.
Ah. That's Francisco's. I see. I'm not sure why. These are not my I'm pretty sure it must be -7. I mean, maybe there is a mistake here. Oh, there is plus. Okay, I don't know how to answer that. If beta F is positive, maybe it's defined in some way that there is another contribution here. Yes. Yeah. No. Okay, but I'm not sure. Yeah, I would have to go to their paper to see how this is defined, and there must be another contribution which makes it overall attractive. Otherwise, it wouldn't work. So basically, to recap,
it's monotonic. That is at least so.
It's monotonic, yeah.
Yeah, but I mean, you have a negative e⁴ minus beta in the
Sorry?
you have negative. Guessing this is the factor in your partition sum. Is that right, or is this something else?
B type is a free energy of bond. But now I'm not sure in this model if it's defined as a single, it cannot be single ligand receptor bond, but maybe it's something collective.
This is, I think, standard. I think particle absorption on
Yeah.
system, the flat surface, the classic natural.
Yes.
Calculate the coverage pattern. So I'm guessing that the two where we have the equilibrium on this.
Mm-hmm. Yeah. Beta. Yeah.
So this is what Dan's paper.
This is Dan's and Francisco Martinez paper. I mean, we can answer that after the talk for sure. Yeah. So okay, this is this paper. So a simple recap of what they saw is you compare surfaces with density of receptors, let's say 2 and 6, so factor 3 difference in number of receptors, and then you take the best monovalent case and you compare the coverage in the first and the second case. It's only 1.8 increase in number of bound particles. While if you then take multivalent particles with 10 possible bonds, then you see a 10-fold increase. So this kind of, if you target now, if you have two surfaces that you want to distinguish, you want to target this one and on this one, you clearly see that you need some multivalent particles to achieve that. So now then we went further to think about the bit more complex problem. So what they showed here is how to target this but not this. That has been answered. Now the generalization of this problem is you have a cell with different population with several different types of receptors on the surface, and now how to target, let's say, cell B but not A or C or any other similar cell. Now we can make a simple theory to study that. Let's imagine we have a cell membrane populated with receptors, C with many different types of receptors. The concentration of each type is Cj, and then we have ligands of population of ligands on the particle, Pi, and we have the interaction matrix between the receptors and ligands. So and then we can, if we assume that, we can write down the partition function. The partition function for the binding of this particle to the membrane is the sum of all these terms, and it's basically the sum of receptor concentrations times the interaction matrix. Yes. Now, if we have a partition, this is for a single ligand, we can then write down the partition function for the whole particle, and we can evaluate the free energy in this very simple model. Now, generally, you try to design the interaction matrix such, so you try to design it so that only the black one only interacts with black, and the red only with red, and blue only with blue. But there is some possibility of cross interactions, maybe with the weaker binding strand, the red one will also interact with black, and so on. So there are diagonal elements, which are probably always the largest, but there are cross diagonal elements as well. So then we wanted to know, here this is the free energy of binding. Yes. Now what we wanted to know, not only how strongly this particle will bind to the surface, but how selective it will be, how selectively it will bind to a specific surface, and not the surface that is slightly perturbed. And basically, what we have to solve is look for the solution, because initially there are several different. If you start with this, sorry for going back. If you start with this free energy, and then you have a given membrane with given the receptor population, and you want to design the particle that will bind here with the lowest possible free energy. This is this is undetermined problem. You have many possible ways to do that. But out of all possibilities for designing this particle, we want to find such which will be the most sensitive to changes in the surface receptor. So we basically want to find the solution with the highest curvature around the minimum, and we can do that with certain assumptions analytically. And I will not go through technical details, but it will give you some simple design rules that come out of this calculation. And the first rule is that if you have a population receptor, the best way to design the particle to be selective is to have a matching profile of ligands. So to each receptor, there should be like a corresponding density. The population of ligands and receptors should be matching. Yes. And then the best way to the best sensitivity will be achieved if the interaction matrix is symmetric. So that's what I said initially. Now these two are kind of obvious, no? Everybody would have thought of it. But the third principle that comes out of correlation is this kind of equipartition principle that all ligands must bind with the same probability. So the probability that black and red and blue will be bound, it should be the same. And that basically translates, this one implies the fourth one is that the individual ligand receptor bond should be extremely weak. And that's what we already saw in the original multivalency study, that you need multivalent particles, and you need all the bonds to be, individual bonds to be weak. So to explore, because multivalency works basically by particle exploring combinatorial entropy. So it's entropy-driven binding. And in order to be able to explore, you need weak bonds that can form and break and form and break, so you can explore. So from our analytical calculation, we get an estimate that the best bond strength is only about kT. And that is in a very strong where it's quite the opposite from what pharmaceutical industry is doing. They want to always find very strong antibodies that bind strongly to the cells. What we are saying now, you should bind weakly, but with many possible bonds. And if you design your system like this, you will have the largest possible selectivity. Now, of course, it's easier said than done. So to design the interaction matrix is very hard in practice. So this is probably more or less what it is. And now, even if the matrix is not symmetric, we can optimize. We can still optimize the process. It will just not be as optimal as it could be if in matrix would be more symmetric. So it's possible to think about designing this matrix, but it involves a lot of biochemistry, and basically these things are not so known. We try to get data for different cells to have receptor population at the surface, and then interaction strand between possible ligands and receptors, and this is it's pretty much not known. Sometimes pharmaceutical companies might have such data, but they won't disclose them. But it's not easily accessible. So if we knew the data for different cells in our body, and for cancer cells and for non-cancer cells, we could design really the perfect probe to target specific cells. But that problem is we need to have this information, and it's not so easy to measure. So okay. Then I said the next question was one question is to target the cell, and then the next question is whether this particle that targets cell will get endocytosed or not in the cell. And here we explored the possibility to increase the super-selectivity by in another way than before, not by engineering the ligands and receptor interactions, but by changing the composition of cell membranes. For instance, what happens? We have ligands that we have and receptors that we have that interact. But what happens if we want to change now the curvature of the membrane, add like curved inclusions or uncurved or whatever? Can we also influence the selectivity of endocytosis by changing the composition of the membrane? Because this, in fact, would be an easier way to control this process than to engineer the molecular interactions. So what we showed is yes, that we can, if we add like particles with spontaneous curvature, they would much more easily wrap the nanoparticle that binds to the membrane, even if they don't interact with the ligands, and even if the overall average curvature of the membrane is unchanged. So we showed that this is the fraction of receptors and the fractional wrapping of the particle, and you see this is without any inclusions, just the membrane and receptors and ligands that interact. And then you see if you add the particles with negative curvature, you can strongly increase this response, or with positive you can decrease. So basically, this is another switch, another control mechanism that you have to the cellular delivery. And this might be what we think, what we speculate here is that this might be the role of molecules like cholesterol in the membrane, as they would facilitate wrapping or the opposite. So that's about this targeting part. I mean, if you have questions anytime, you can ask me, but now I'm going to talk about another topic. also that is also connected somehow to multivalent binding, and this is about activation of receptors in the autoimmune response. So this work was done with in collaboration with Jure Dobnikar at UCLA and his lab, and with the group of in Lausanne Hospital immunologists and with Dan Frankel and Tine and me. Here, first a little bit of background. That is like a good simplified picture that I can understand of how immune system reacts. So this is like one way of response, which is mostly adapted in innate immunity, is this plasmodendritic cells that contain different receptors that are called toll-like receptors for some reason. And this, for instance, toll-like receptor 9 is in these cells responsible for detecting viral or bacterial DNA that enters the cell. So, for instance, virus comes, we are invaded by the virus. Virus is internalized in the cell, then disintegrates, and this viral DNA is detected by toll-like receptor. And when this happens, this receptor is activated, and through some downstream reactions, it triggers response, which is production of interferons alpha. And this interferons alpha then go around the body, signaling different kind of response to the other cells. This is called the entire immune response. Basically, the cells stop reproducing to not let the virus hack them and reproduce itself. They go and create other apoptosis to certain cells with viruses and so on. I mean, there's a complex immune response. But what we were interested in this work is this first step, how this is activated. So this is a 3D structure of this toll-like receptor. See here, and this is DNA. So you see what happens when the viral DNA comes. This toll-like receptor here has a strong positive charge in the center, and this charge, this site can interact very strongly, but it's base pair sequence specific. So it interacts very strongly with CG sequence that is quite often present in viral and in bacterial DNA. And therefore the microbe DNA will usually bind strongly to this receptor. While our own DNA doesn't have this kind of sequences, not in similar abundance as the viral. So that means that our own DNA will not bind to this receptor and will not trigger autoimmune response, which is how it should be, because otherwise we would always be ill. No? So self-DNA there is no activation. That's the design. And but no, then what was discovered, what happens is that self-DNA with certain antimicrobial peptides can make complexes that do cause this activation and trigger immune response, and that is basically autoimmune disease. So we think we are attacked, but it's basically that complex DNA, self-DNA coupled with antimicrobial peptides, they are detected as an intruder, and then we have this reaction. So it has been established that this peptide called LL37 is doing that quite efficiently, and this is known for some time, but it hasn't been from 2007, let's say, but it hasn't been there was no explanation, no understanding why LL37 is doing that and why other peptides are not doing that, and how does it work. Yes. So what Gerard did in his group, he took several different peptides and in solution and let them form complexes with DNA. And then he took these complexes in X-ray experiments and determined their structure. So basically what happens is that they form finite size clusters of about, this is the whole size, about 5, 6 times 6 or something like that, that have a liquid crystalline symmetry. As DNA here only DNA is plotted, but not proteins are in between. So for clarity, it's only DNA that is plotted. So, and you see that DNA can be very condensed or expanded, and it could be in square or hexagonal arrangement, or even other symmetries. And they did this for several, like 20 or 30 peptides and DNA combinations. And then they produced scattering plots, and from that you could see this lattice parameters. And now they had all these complexes characterized, and then what they did is they sent it to the immunology lab, and they injected those complexes into plasma dendritic cells and measured the interferon production of the cells exposed to these complexes. And what you see here, these are some of these complexes, and this is the interferon production. You see that, and this axis is the first peak position, which means the lattice spacing between inverse lattice spacing between DNA. And what you see that there is no, for very small lattice spacing, there will be almost no signal. And for very large lattice spacing, there will also be very little signal. Well, for like a very specific lattice spacing, all the peptides that are able to make complexes with this specific lattice spacing, they produce a large signal, yes? And that there is this sweet spot spacing between DNA that seems to do the job. And that's when we started talking to Gerard, and we made this simple model. Basically, if you look at the structure of a toll-like receptor, it looks like that. Has a positive charge in the center and a slightly positive charge also on the backbone. While DNA is a negatively charged rod. In our simplest model, this is just a charged cylinder. Now, you have a single long DNA. Then the average binding energy of this receptor to this DNA is just the parameter epsilon in our. Basically, what we are doing here is a Langmuir adsorption model. We assume that there are m available sites on the DNA, and NB is then the number of bound receptors. And we know that this NB over M is given by this expression. This is a Langmuir adsorption. But now we don't know exactly what is this epsilon. But if we believe the experimental information that for one single human self DNA there is almost no binding, then we know at least the limits of this epsilon. Epsilon must be of the order of, like, say 4, or let's say we assume that there is NB over M must be less than a few percent, and that gives epsilon must be a weak bond that comes purely empirically from observation for cell DNA. For viral DNA, because it has specific system sequence, this epsilon will be larger, and then you will have binding even on a single DNA. But for human DNA, epsilon must be something like that, 4 or larger. And then now, what happens if you have not a single DNA, but the lattice of DNA with some lattice spacing? Then it happens that the binding happens here, and also there is additional electrostatic interaction between backbone and the next DNA in the lattice. No? And this additional electrostatic attraction, because it's positive and negative, renormalizes the binding constant. It makes it stronger. And we assume a simple DLVO expression for the electrostatics here with the correct, like relevant, reasonable parameters. And what we see is that now the binding, effective binding becomes very different. Yes. So here, if it was 4 before, and now if it changed here, this is A is lattice spacing. If it's infinite, it's like a single DNA. Let's say we are here somewhere. We believe we have to be around 4 or 5 for our system. But then we change the lattice spacing closer and closer, and we see we go down. We can go down almost to -4. No, so it's a huge increase of binding strength. Just because of the geometry of the self-assembled cluster. And then if we can make now for the lattice, we can make another prediction for the partition function. Yes, we have to explore also the fact that there is a limited, if you bring lattices closer together, there will be less possible binding sites. No? This is another effect we include, yes, because of the steric effects. But even with that, what we can show is the number of bound receptors to the cluster has this dependence, and in a certain limit, this becomes a double exponential. Yes, it has a very strong dependence on the binding energy and through this on the lattice spacing. So so this becomes very sensitive to the lattice spacing, and basically sensitivity, selectivity parameter that we can define. This is selectivity parameter, which is number of active receptor is the receptor that is bound, because then it changes conformation, becomes active, and then it can trigger the immune response. So number of active at the given lattice spacing divided by number of active at the infinite lattice spacing is the selectivity parameter that we define. No? And this selectivity parameter from the experiments that we had before, we could estimate that it is of the order of 100, let's say. And from our simple simulations, we could also confirm that this is of the same order. And basically what we observe is here is the binding number of bound receptors as a function of Q, the inverse lattice spacing. You see this behavior for different bindings, individual binding strands. And this is number of all total active receptors for a complex. And you see this is logarithmic scale, that we have this strong preference for a specific binding for a specific lattice site, which very much agrees with the experimental. I mean, it's it's consistent with experimental observation. As it's a very simple model. So, but we believe that this is actual explanation, that geometry of the self-assembled clusters is crucially influencing the multivalent binding then of the receptors to the cluster, and then the activation as a consequence. So that is one thing, and this mechanism could be present in other systems, like it could be responsible for some types of allergies, when it's not necessary that you're allergic to some substance, some particles, but maybe you are allergic to the complexes these particles make with your DNA, and this could be further explored. And also the same is to decide why some nanoparticles are toxic and some are not. It could be that the nanoparticle is toxic in itself, or it could be that the complexes made by peptides or by DNA are toxic. So that is something to think about. We also had ideas about this could be used in treatment of the autoimmune disorders, because this what Gerard studied were the autoimmune disorders like lupus and psoriasis, and there is no nice cure for that. There is no efficient cure. The medicine that is available doesn't work well, and it's not easy for patients. So we thought that if we understand this, and if we can like outperform the LL37, which causes this problem with some simple molecules that make slightly different complexes, we could not cure the disease, but we could stop the symptoms. Apparently, this work is already like 8 years old. Apparently, they were thinking about doing that in clinical trials in UCLA, but I never heard of any progress on that. So it's probably more complicated than we think here. It's also another aspect of this is we then later saw the same effect in different systems like RNA peptide complexes activating total like 7 receptors. Is pretty much the same story. And then also in the program cell death, NETs, nuclear core complexes that are expelled from the cells, like these are nuclear core complexes that are expelled from the cells. Then they form stacks and clusters. But what happens is, what is measured is that this pitch of DNA that is wound around the core complex in the extracellular environment, it changes. It becomes larger. You see here, it becomes close to 3.4 nanometers, which is exactly the sweet spot spacing that we had in our previous system. And not surprisingly, now to us, these nets invoke a strong inflammatory response of the body. And what we believe is that this exact pitch, again, is able to bind toll-like receptors that then they trigger autoimmune response, which makes cell death inflammatory. This paper is still in progress, but it's a very convincing observation. Okay, so I can skip that. And just as a very short comment, we also explored multivalency in the problem of targeting a genome. So basically, the detection of bacterial genome works like that. Usually you have some surface decorated with probes. Probes are like single DNA with a specific sequence, and then you take the genome of blood sample, you fraction the whatever is inside, and then you look at how much of these short segments in the solution will bind to this surface probe. That is one way to detect and then you do the amplification and all this. But we have showed like we did simulations now with exactly this approach. If we change the probe surface density, the specificity of our detection changes like this. But then we were sure that we can design a better system. So if instead of short segments you take the whole long DNA of you don't segment it, and you designed a better probes that we made a whole algorithm of how to design optimal probes for that. Then this DNA genome can now multivalently bind to the surface, and we have shown again that by doing this, by optimizing this procedure, we can have a specificity which is like orders of magnitude better than the current methods now. And this is something that works very well. We could, let's say, have the surface which is decorated with the specific sequence designed to recognize E. coli bacteria. And this is how it looks when you put E. coli genome. And this is how the same surface looks when you put Bacteria subtilis genome. So it does recognize E. coli and distinguish it from, but we can even design sequences that selectively binds different strands of E. coli. Yes, because you know the genomic sequence, you can design this. The only thing which we are still working on is how to detect viral genomes, because bacterial genomes is easy because it's long enough. It has many repeated sequences, and it can easily form multivalent binding. You can design system to be in the regime of high specificity. Well, the viral genomes are usually 10 or 100 times shorter, and then it becomes like a very tricky to design it so that there is enough multivalent binding. But we are working on that, how to solve this. There are some results, but not yet finished. So that would be all. I hope I showed that multivalent bonds multivalent binding is really a crucial mechanism that you must have if you want to have a selective targeting. Yes, and that is true in biology. It is used this fact is used in biology, but it's also true if you want to design any artificial system that will have high selectivity. You have to think about multivalent bonds, not a monovalent. So we can design carriers that will target specific surface with specific composition. Theoretically or in simulations, it's easy to do. And if we know the data from microbiologists, from biochemists, we could design very specific carriers to target what we want. And it also plays a role in activation processes, in biological activation. And one example I presented, but we believe that this is more general, that basically specific interactions in biology or in biochemistry are all kind of the consequence of multivalent bonds. There are no specific interactions, fundamental interactions. Electrostatic is not specific, but because of either geometry or entropy, the higher order interactions can become specific, and the onset of specificity is, I think, explained by multivalent interactions. And I talked about novel genome detection strategies. But thank you very much.
Okay, so thanks very much for are there any questions?
Thank you very much. Something it might sound stupid, but this is your last piece of probe. Can you read it to kind of the next question?
So
mRNA.
So our idea is to use this we started this project. This is basically Rosalind Allen was the leading scientist on this project, but it involves a lot of people. So the starting idea is to design some method to detect genome, bacterial or viral, in a very simple way, very cheap way. Also, our idea here is to put these probes on the colloid, and then if you have genome, then the colloids would aggregate, make a large cluster, and a cluster that would change the turbidity of the sample, that you could just look by eyes and see if it's bound or not. So that would be a huge advancement of how to detect bacterial infections. As actually, until recently, it didn't work quite well because these colloids would cluster. I mean, it's not so easy to make them not cluster in any way. But recently, Eric Eiser and a student postdoc in Beijing, they did experiments that exactly prove this, that this is possible, and it works for bacterial genome. It actually works. Now, we want to do it also for viral genome, and this is a bit more tricky, but I think it might work as well.
So, in the design, the first part, so these ligands and receptors, you only consider purpose, although there are some design which allow the function between different.
Yes. I mean, one problem, if you talk to biochemists, is this binding strength that we say it has to be a 1 kT. Most biochemical bonds are stronger. Yes? So one way to make them weaker is to have some repulsion, some non-specific repulsion, for instance, coat the surface with polymers that will have some repulsion. Sometimes you want, I mean, depends what, this is a bit of a game, no? If you want, you also want a range selectivity. That was Stefano Angioletti worked on that. You can design system with combining repulsion attraction that it's sensitive to a range of concentrations and not too smaller, not too larger. Yeah? So you can, design whatever what is your goal. You can use this to design the kind of response. Yeah. It's possible that in some cases this would increase selectivity, but it's not. So one thing that is also important to consider is it's not only. It's not only the binding selectivity. I mean, it's here we have maximized the selectivity, which is fine. But if at the same time your overall binding is very weak, then it doesn't really help if it's very selective because it won't bind. So you have the other constraint that the total binding has to be strong enough. No? So this is just a theoretical exercise to see which is the most selective, but in practice, when you design that, you have to also have binding, enough binding. So the constraint optimization must be done for each particular system.
Jure, can you say a little more about, you showed some pictures of a nanoparticle going through, and then and these were simulations. And so can you say a little more about the simulations, or were they atomistic?
It's this is a membrane model, one particle layer membrane model, which is used by Angela Šarić and her group, then we have in this membrane we have these receptors that interact with ligands on the particle, and then we have membrane particles which are inert, and then we have inclusions. Okay, but that's not important. So the simulations basically take the nanoparticle, then it binds to the receptors, then the membrane it wraps around. Now we don't measure it going through, we just measure the amount of wrapping of the particle. And we say if it's fully wrapped, it's through. Yes, so that's our.
And can the membrane be persuaded to break and let the particle into the cell?
Yeah, I mean, we don't do this so far. We just calculated the surface area fraction of particle that is attached to the membrane. But yeah, I mean that's what happens. Then it breaks. Yes, and yeah.
So you want to see a system where there is lipids all around.
Yeah.
And the particle is kind of sitting there.
That's what happens. That is work of Robert Waha and Dan also and Gerhard Gomper, and that's what happened. But I mean, our membrane model is not that realistic. I mean, we do not want to study really dynamics of endocytosis. We just wanted to see how selective would that be if we add inclusions.
Okay, I had a question.
He has
No. So about your immune part, when you spoken about those proteins, they bind bacterial DNA because of specific sequences which are often there.
Yeah.
And human DNA lacks this specific sequences, so it doesn't bind. And your theory was purely electrostatic. You should work for anything bacterial.
No, that was measured actually in another paper. I didn't put these results here, but they measured. Where is it? They measured the binding of receptor to DNA with sequence dependent. I mean, they have really done a lot of. And it's clear that there is a much increased binding for, I think, CG is a sequence. And this is then present in much more abundance in viral and bacterial DNA than in human. It is practically not present in human. I mean, that's.
Static theory misses this effect.
It's not electrostatics only. It's more than that. No, it's confirmation. In my theory, it's just electrostatics, but this epsilon will now be different if you want to describe virus. I mean, we don't have specific sites. We just have one epsilon for the whole chain. But if we want to now simulate what happens in the bacterial or viral DNA, we would use a stronger binding epsilon, and then you would have binding to a single chain. Well, in human, it's weak enough so that it doesn't bind.
Okay, so I think I'll ask you.
Okay.
Thank you very much.
Thank you.